Okay, let's start. It's time. Okay, so today I'm gonna walk you through a few vignettes highlighting which something I think is important to study modern cell biology, and I've already touched upon those things a little bit in the cell migration lecture. So mostly we're going to go through specific examples dealing with these so-called biosensors. So how do you visualize and quantify specific activities in the cell? How do you manipulate them through optogenetics? And at the very end, I'll touch upon one example of a so-called synthetic biology where you can create a biological system from scratch. But that last part is mostly for information. I'm not going to ask you any question in the exam about the very last part on synthetic biology because it's a bit of a moonshot anyway. Okay, so first off, I'd like to highlight some of the key problems that sort of led me to want to tell you about these things today. So throughout the course, we've seen that a lot of the cellular activities, protein activities, happen not at the level of the whole cell, but in very defined times and places within the cell, right? Be it at the membrane, at the ER, etc., etc. And they also happen over very short period of times. But, and in all cases, you have, so if we take the example of actin regulation, for example, we've seen that there are pools of actin, pools of regulators, of effectors that are acting in different places in the cell. Right, so I already alluded to this idea before, but if you take a mutant cell, you're going to deplete the whole cell at all times from the protein, and so you may not be able to interpret the phenotypic outcome in very accurate terms. Especially, you won't have the correct spatial and temporal resolution for your assay. So that's what this lengthy text is saying. And actually, with one of my postdocs, I wrote a review talking about this quite a bit a few years ago. So, so to kind of deal with these questions, that's why I wanted to introduce to you these tools, because those of you who might continue in research and so on, if you're going into cell biology kinds of research, you might want to think about these sort of tools which are really state-of-the-art and revolutionizing the way we do cell biology. So so we're going to start talking about these biosensors. I introduced to you the other day FRET, so this fluorescence resonance energy transfer, or sometimes called Foster resonance energy transfer. We're going to see examples of so-called bimolecular fluorescence complementation. This sounds complicated, but you'll see it's fairly simple. And then as we go, so this is more descriptive, and then we're going to look into tools to interfere with protein activities with greater and greater spatial and temporal control. And depending on the time, I'm going to tell you about this sort of synthetic biology approach. So let's start with these biosensors, both FRETs and bimolecular fluorescence complementation. So this first slide here must somehow be familiar for those of you who were here on Monday. No, I haven't uploaded the recording yet. I will do that today. And so the idea of FRET, the fundamental idea of FRET is if you have two fluorescent proteins, or fluorescent molecules, so lately fluorescent proteins have been used a lot. And here, so what's really important to understand is you need a pair that matches relatively well in terms of absorption of light and emission. So here we go. So CFP, cyan fluorescent proteins, they tend to absorb low wavelength photons. So for example, this 433 nanometers. So this is in the blue or UV scale. So this is usually high energy, right? Within the whole visible spectrum, it's among the high energy. You're all familiar with that, right? Low wavelength, high energy photons, higher wavelength, lower energy. Okay? So, and so you must also be familiar with the idea that so if this is a photon of a certain wavelength that's absorbed, the emission is always at a higher wavelength, lower energy, because there are always losses, Okay, you've seen this concept when you talked about energy transfer in the metabolic, in the electron transport chain, for example. You always lose some energy. Okay, so you can always emit light at a slightly longer wavelength, so slightly more towards the cyan here. So UV is absorbed, cyan type light is emitted. Okay. Now that's if there is no so-called FRET acceptor. When there's a FRET acceptor, so what does that mean? That means here you have a YFP, and it's so close that you can have transfer of energy. The energy that's been absorbed from this photon is being transferred without emission of light. Okay, so this needs two things: compatibility in terms of what the acceptor, the amount of energy the acceptor can absorb. So it must be compatible with this, give or take, and close proximity. So when this happens, you can have FRET. Okay, and then the way the system will go back to its initial state is now the YFP is going to emit light. Okay, so the important point is that normally, if you shine 433 nanometer light on YFP, it cannot absorb it efficiently. Okay, it's too low. A YFP will be excited by wavelength in the order of 480. So usually in labs we use 488 nanometers. It's a classic wavelength used to activate. Actually, YFP is 514 nanometer. That's its preferred, its maximum absorption wavelength. Okay, so this is much lower, and here it gets the energy through FRET. Okay, so what I told you on Monday, and that's true here, is that all the FRET-based assays, they rely on this transfer of energy. So they measure light emitted, and you quantify it with regards to how much light was shone on the system in the first place. But what they are trying to get at is the proximity of these two proteins, which will be a proxy for the proximity of the two proteins they are attached to. Right? We saw this, that was the principle of the Rho GTPase reporter we talked about on Monday. Okay, so this general principle is used a lot in cell biology to make these biosensors. So here is one example where you see the FRET signal. So it's always expressed in relative terms. There is no absolute value for FRET, right? It's always relative. So you can always study or understand what's going on with regards to a reference. Okay, there's no absolute measurement of FRET in this case. Okay, so in other words, the amount of FRET is the amount of 530 lights you get per 433 light you're shining on the sample, right? Give or take. So what I'm going to do in this lecture is every time give you kind of the general idea and then illustrate it with bits and pieces from the relatively current literature. So here is a paper, every time you have the simplified citation here, from Denise Monter's lab in Cell a few years ago. And so this lab studies cell migration using a particular system that's the fly ovariole. So the fly ovary or the egg chamber. So what you're seeing here is an egg chamber of a fly, Drosophila melanogaster. Here's the prospective oocyte. Here are nerve cells. All of this is the germline. And here there are follicle cells, which are somatic cells. You can write that down if you've already registered for developmental biology next semester, because we're going to go through that and you're going to have to remember that. And I know some of you have told me already. I know, they told me. They're like, oh, we registered for your class. I'm like, so write that down, because this is going to be there. So these cells here, the border cells, they have been a really good model for collective cell migration, because so in the egg chamber, so this is a so-called stage 10, so it's fairly advanced, but it comes from this early stage, stage 9. And in the fly ovary, you have many of these egg chambers developing in sequence. When you isolate a fly ovary, you have many ovarioles, and one ovariole is a string of such egg chamber at different stages. Okay, you have early stage and progressively later stages. So from the same oviole, here you have another egg chamber that's just slightly less advanced. Okay, and what you see from this is that you have these cells here, the border cells, so they have just delaminated from this epithelium. And here they are at the back, if you will, of the egg chamber, and they will actually migrate forward until they reach the oocyte, which at this stage is still here. It will grow a lot between that stage and that stage. And now the border cells are here. So these people are using this model to study the migration of these cells. Okay? Now, in this paper, they are studying the activity of E-cadherin. So we had to skip the cell adhesion lecture, but cadherins are so-called homophilic cell-cell adhesion molecule. So in other words, you have the same molecule, it's expressed in cell A and cell B, and they like each other from different cells, so they help cells bind to each other. It's so-called cell-cell adhesion. Okay? So this cadherin is a transmembrane protein. And the idea is that you have cadherin, so you see here cadherin in green labeled with an antibody, so it's strongly expressed in the cells which have migrated. And here in this experiment, they used, so this is complex flight genetics. So how many of you are doing some internship in fly labs? There are a few fly labs. Is there anybody? Nobody? You are? Yeah, that's right, Claude. Okay, so you may be familiar with this UAS-GAL4 system. It's a transgenic system to express recombinant proteins in the fly. So here, they are expressing GAL4 specifically in these cells using a promoter from slow border gene. I'm giving you a lot of information you don't need to remember. And it's used to activate expression of this construct here, which will cause RNAi. You know what RNAi is, right? RNA interference. What does that do? It leads to degradation. Well, interferes with RNA. Okay, thank you. It leads to degradation of the RNA when it works well. So that means what happens in this experiment is that you have a specific degradation of the E-cadherin RNA in those cells here, right? And so you see here now they have not delaminated, they have not migrated, it controlled they are here, so they should be located in this position. They are still in the epithelium in the back, and they do not express high levels of E-cadherin. So this is the first evidence for what? Come on, biologists. That you require E-cadherin in these cells for them to migrate. Right? Simple experimental study. Okay, so that was the simple part. Now let's get into the complicated part, and the reason why I picked this paper. Because what they wanted to do, so cadherin, right? So you have cells that migrate. This is a protein that's involved in cell-cell adhesion. And migration, you're going to have force involved, right? Pulling, pushing, and so on. So what they wanted to see or to try to visualize and even measure is how much force was applied onto cadherin, and if there was any pattern to that. So they developed this tool based on fret, right? So here the idea is to use fret to measure tension. So how much force is pulling on a given protein. So here's the idea. So you have this sort of spring here, which is actually encoded in a protein. So it's, a sequence of amino acids that will be able to act almost like a spring. And on the N-terminus, you have turquoise fluorescent protein. So that's similar to a CFP, and Venus, which is a YFP, right? So everything in place for fret to happen. The idea is that when there's low tension, you have this spring is sort of coiled onto itself. The two proteins are close together. You can have high fret. Now, if you place this system under tension, especially by pulling on both the CFP, or the TFP, turquoise here, and Venus, you increase the distance, so you have this spring is not strong enough to resist, the tension, so you increase the distance between the two, and fret goes down. Okay? Yes?
Is this spring naturally in the protein?
No, this is all engineered.
Okay.
Yeah, it's all engineered. And they probably had to do quite a bit of trial and error to get the right physical property for it to be relevant. Okay?
It's like an alpha helix?
It could be, but I'm not entirely sure it's a alpha helix in this case. Yeah. Yeah, I don't know exactly what the structure is. I doubt it's an alpha helix, actually. It's probably bigger than that. But, so the idea then is they created this transgene, this so-called E-cadherin tension sensor, by placing the spring on the C-terminus, which is in the cytoplasm. Right, so this is a cell-cell adhesion molecule. So you have an extracellular domain from one cell, that's the plasma membrane, so you have one cell here. The extracellular domain binds to the extracellular domain of the same protein in a neighboring cell, because that's what I meant when I said it's a homophilic adhesion molecule. That means it likes to bind to the same molecule in the other cell, okay? So when you have the same molecule in the other cell, and very much like what I said on Monday about integrins, for cell-cell adhesion to happen and to be mechanically functional, what do you need? Coupling, mechanical coupling with the cytoskeleton of the cell. Okay, so this is all achieved in the case of cadherin. So this is a very well-studied system, okay, in mammalian cells and so on. So it's mediated by this beta-catenin and alpha-catenin, which you may have seen or will see. Beta-catenin is involved in signaling as well. But here this is a structural protein that links these cadherins to the actin cytoskeleton. So you have mechanical coupling between the cell-cell adhesion and the cytoskeleton. So now you have everything in place. You can have some tension at the level of cadherins, and the whole idea is that this fret system is going to report on the tension, especially on differences between different parts of the cell. And I'm just going to show you, not the whole paper, but just this snapshot here. So here is how it works. So this is a control. So that would be a case where you have the fret system, but it's not engaged in cadherin or anything, right? So it's just showing you have high level of frets sort of everywhere at the membrane. That means the default state is the high fret, okay? So what now they are going to look at is lowering fret will report on higher tension, okay? So this kind of gymnastic, I want you to be able to, have that. And so here's the idea. Now that's the real cadherin tension sensor. So red is high fret, blue is low fret, and green is kind of intermediate. And here, so this is a cluster of cells. It's not so easy to see. And we know it's moving in that direction, from the left to the right. And actually this here is a big protrusion from the leader cell, right? So what basically you can see is that here at the membrane, you have very little red and a lot more green. And here, you have a lot more red, okay? Simply put. You can quantify the ratio, so-called front to back, so how much tension is there or how much fret is there in the front relative to the back. And indeed, you have about 10-15% less fret in the front than in the back compared to the control. So what does that mean in terms of tension? Higher tension where? In the front, absolutely. So that was one of the discoveries of this paper, is that and one argument to say, well, indeed, in this case, the cell is protruding, and in the protrusion, you have cadherin engaging in cell-cell adhesion contacts with surrounding cells, the nurse cells actually, and that is used to sort of pull, right? So you have greater tension in the front than in the back, which was not formally known in this system, right? So that's one illustration of how you can use fret. Yes?
How is the control different?
So the control is different in the sense that it does not engage in cell-cell adhesion. It's at the let's say you could. I'm actually not entirely sure how they did it in this paper. I looked up. I didn't look it back, but the idea is that if you create it in a way that you lack, for example, the extracellular domain, it's going to go to the membrane. It's not going to engage in cell-cell adhesion, so the default state will be the high threat. So that's, in these experiments, you always need such control to give you the dynamic range, right? So just as a reminder, on Monday, when I showed you the threat for the raw GTPases, there's this plot. There was a busy slide, arguably, but I showed you the level of threat you get when you have a constitutively active, so it's like maximum all the time active, and the level of threat you get when you have an inactive GTPase. So the difference between the two is your dynamic range is the range that will be informative in the experiment. Okay, so in a way here the control is important for that. All right, so that's the biomolecular sensor. So now, as emerging experimentalists for some of you, what could you imagine would be a burden here? Have you guys done a microscopy, fluorescence microscopy? Not so much. Now you really want to do it, right? Well, usually, when you do fluorescence microscopy, you don't have an infinite number of channels you can use, or colors. Yes?
I was just going to say that time would be an issue.
Sorry, say that again?
I was just going to say that time would be an issue because,
Time?
Yeah, if it's migrating, has different tension.
Yeah, so this is a good question. So actually, yeah, it's a pretty good question. The scales, though, are very different. So this type of imaging can be done really quickly.
Okay.
And so it's the order of seconds, if not milliseconds, in the speed at which we can image now. But the migration itself takes hours. So when you're looking at integrated cellular processes or developmental processes, usually it happens over timescales, the order of minutes or more. But these molecular events happens on timescales which are the order of seconds, if not milliseconds. So you can actually image quickly and get a lot of information, and yet the system has not completely changed. That's a good question. So my transition was, here you need to occupy two channels, two different colors, right? And so people have developed ways to use only one color to still report on these protein-protein associations. So this is the principle of bimolecular fluorescence complementation. And I'm just going to show you this one example here. It's very simple. So GFP or many fluorescent proteins, they have this sort of structure. So how would you call that? A beta barrel, absolutely, right? So beta sheets, and this would make a beta barrel, and in a way, this structure is responsible for the ability of the protein to absorb light of a given wavelength and emit light of a higher wavelength, okay? So the idea of complementation is it's depicted here and here. So you have 11 beta sheets like this, okay? Yes? My mic turned off. Well, there's no battery. How can I work in this condition? Thank you for letting me know. Can you guys hear me in the back? They're like, yeah, unfortunately. Yes. Yeah, because it's not going through this at all. Yeah, it's only going through the computer. Okay. Maybe this guy can help me. Hello?
No.
Hello? Man in the box. Okay, I'll continue. So, bimolecular fluorescence complementation. So the whole idea is that you need these 11 beta strands to build the beta barrel. So if you take away one, it's not working. So what people have done, and this is very clever because sometimes one problem when you attach a fluorescent protein, so GFP is in the order of, let me remember, something a bit more than 200 amino acids. So it's not very big, but for some proteins, it may be big enough to interfere with the processing, interfere with the function. So the idea here is to attach only the 11th beta strand to the protein, right? So that would be for GFP, it would be this short stretch of amino acids. And then express in the cell the rest, so the GFP with the beta strand 1 to 10. So alone, neither of those can fluoresce. But when they get together, and they tend to spontaneously, now you can have fluorescence. So it's one way where you can actually have a very small tag on a protein. And for genome engineering applications, it's useful because it's easier to integrate a small tag than a big GFP coding sequence in the genome. And then you can express this FP1 to 10 and still recover the pattern that you would expect. So I'm just going to show you some examples. So here is beta-actin. Beta-actin, right, found in actin filaments and so on. So not surprisingly, all over the cell, you see these filaments. And now you see it very nicely, although you've only labeled actin with GFP11. Okay? So presumably interfering the least with the dynamics of the protein, because it's a very small tag. Same with cherry, and here I'm just illustrating some examples from that paper, where you can literally take almost any protein in the cell, tag it with the GFP11 beta strand, and as long as you express GFP110 in the cell, you're going to get fluorescence where the protein is. Okay? So here the examples are self-explanatory. Clathrin, actually, where would you expect them? Clathrin. In vesicles? In all vesicles? It's like, yeah, I dreamt about it. There was AP complex, there were vesicles and clathrin, and right. So receptor-mediated endocytosis, trans-Golgi network. So at the packaging stage, right, it helps make the vesicle. Now histone H2B.
Does it always spontaneously come together, the 11 and the 1?
Yeah, they kind of do. I mean, so I know it's not so easy to visualize, but because it happens at the molecular level. So you have Brownian movements, and those are still small proteins, especially the GFP110 is still small. So it diffuses freely in the cell, and the diffusion rates are relatively high. So very quickly they are everywhere. So the chance that it will encounter the GFP11 is really high, right? And then when it happens, because it's part of the same structure, they tend to fold together. Yeah, but that's a good question. So anyway, histone H2B, chromatin, beta-tubulin, microtubules, zixin. I've not told you much about this. It's in cell adhesion, so integrin-based adhesion. So you see these dots here. Calreticulin, ER, and this one beta-2 AR. So that's a transmembrane receptor. Okay. So this is very descriptive. Now let me show you an example of a sensor based on this bimolecular fluorescent complementation, which is really powerful. So it's a very famous sensor in neurobiology called GCAMP. And the whole idea is a variant of this BFC or split GFP concept called circular permutated GFP. So what is that? So here they are not splitting beta strand 110 versus beta strand 11, they're basically splitting GFP in half, N-terminus versus C-terminus, okay? And what they do is they clone it instead of having N-terminus first and C-terminus next, they clone it in the opposite order, right? So the C-terminus is cloned first in the protein, and there's a little linker between the two. So, and more importantly, it's attached to these two domains here, which are sensor for calcium. So when you have high concentration of calcium, these two domains come together as depicted here. And the conformation is such that now the GFP goes back into its fluorescent conformation. It's fairly complicated at the molecular level. I'm going to show you a molecular model next. But the idea now is that you have GFP fluorescence, which reports on calcium concentration. Okay, so that's very interesting because imagine calcium concentration in cells is not easy to see, especially live cells. So now that's possible because you're going to have more or less fluorescence based on the concentration of calcium. So this protein has been around for a while, and people have done protein engineering to improve its properties. And so here is a paper from a few years ago now showing further improvements, so specific mutations in the calcium binding domains as well as mutations in the GFP. So the CP stands for the circular permutated GFP. So now that would be the structure of the protein bound to these 4 calcium ions and the GFP back into its fluorescent conformation. Okay? I mean, just for your information, what's important is that when you have an action potential, so now this is in a neuron, okay? So you've seen that with Claude recently, I believe. Action potentials. I know it triggers lots of good memories. And so the idea is that when you have an action potential, what happens with the calcium concentration in the cell? Tends to go up, right? So by and large, calcium concentration in neurons is a decent proxy for neuronal activity for action potential. So what's measured in these plots is, so this delta F over F0 is, you have a baseline fluorescence, because this sensor is not non-fluorescent, right? There's a baseline level always. What you're looking for is an increase. So the delta F is the difference in fluorescence relative to that baseline. Okay? So you're measuring. Constantly, you have your baseline, and then you can now measure the difference. And what this is showing you here is this GCAM6S performs really well because so the 0 has been set at this is when you have an action potential. And here you see with only one action potential, you have an increase in about 30% fluorescence over baseline, and then it quickly returns close to the baseline after a few seconds. Okay? So what this is illustrating is that you have a sensor of calcium concentration which will report quite faithfully on action potentials in neurons, in live neurons. Okay? So let's use this, or let them use this. So what they did in this paper is express GCAM6S in neurons of live mice. And basically, when mice watch this grid pattern, some neurons, so the researchers can change the orientation of the lines, okay? And some neurons will respond to certain orientation or others. So for example, the color code here is that the neurons, so here you have field of neurons, right? So you see these yellow bodies, blue, red, and so on. So the color code is that the red, they will respond to grids oriented at 45 degrees. Okay? The blue ones at 90 degrees, 135, and so on. Okay? So what they do here, and they record, so they measure the GCAM fluorescence in these 3 neurons. One, so that's a blue one, so it responds to 90 degrees, red, and this one, which is green. So you see, they start, so here are the 3 neurons, this is time, and this is the change in fluorescence. Okay? So first neuron, they show a graded pattern in this orientation, no response. Because the first neuron here is blue. So blue, boom. You show this pattern. Now it's horizontal, 90 degrees. Now this neuron spikes. Okay? So So I'm not making a good job in explaining this, but you should be really impressed at this point, because that means people are starting to be able to visualize which neuron responds to exactly what pattern is being shown to the mouse. Yeah.
Can you describe the pattern a bit more clearly?
So this is the pattern.
So
It's
just
a grid.
a sequence of lines that
It's
go
a-
in a direction.
No, you just show it a grid, and then you change. You show it a grid oriented in this way, and in this way.
So it doesn't move. It just.
No, I don't think it moves.
Okay.
Well, I mean, the movement is it appears or disappears.
Okay,
Yeah. So let me show you movies then. So here what you're gonna see are the spikes, so the action potentials themselves, and the response of GCamp in the cell body. So this is the cell body. See spikes? So it's not easy to see, but here you see the quantification of GCamp, and it matches pretty well these action potentials. Right, so here we don't know exactly what is being shown, but what you're seeing is that you have a very good correlation between the spikes of activity and the spikes of activity of GCamp, the reporter. And so now for that, you don't have to patch the cells, right? So for action potential electrophysiology, you have to patch. Here you just have to, it's less invasive. You just have to use the microscope. Yeah, that was a big one. Okay. Now, that's at the level of the cell body. So neurons, they integrate a lot of information at the level of cell bodies, but the individual inputs, they come at the level of dendrites. Okay, so whether, sticking out of dendritic spines, I should say, so sticking out of dendrites or even on axons. And what this movie is illustrating is that now you have a dendrite, and you have several different spines. So spines, these are little protrusions, and this is where you have synapses with different neurons. Right? So each neuron has connections with hundreds of other neurons. And those synapses are specialized in some way. Okay? So what this movie is going to show you using GCamp is that individual dendritic spines respond to particular orientations. See, this orientation activates these guys. This one responds to the horizontal. I know you all seem a little blasé. I know since this you've now seen a black hole, and it's like, okay, whatever. Okay, so far all of this is, I think, pretty good, but observational. Right, so we have great tools to assay what's going on. And once I told you, when you do experiments, you need at least two things. You need an assay, something you're going to measure, something you're going to observe. So with all these tools, you can now refine observations, refine measurements, but we have not talked much about perturbations. So something you can now do to the system. How can you control activity? And if you think back about my introduction, there were two levels of resolution we wanted to improve, right? Compared to, for example, a mutant. So a mutant cell is lacking the proteins from the beginning at all times everywhere. So ideally, even if we want to achieve a loss of function or a gain of function, we would like to control the time at which it's active or inactive, and even better, the time and the space in the cell. Okay, so let's see how we can do that. So the first one, I'm going to tell you about controlling the time. So here, this illustrates a pretty cool system that's been developed, again, a few years ago based on targeted degradation. So it's called auxin-inducible degron. So what does that mean? I mentioned, and I don't know if you've had a lot of information on the proteasome, the protein degradation complex, right? So you know it exists. You know the cells have ways to degrade. I know I mentioned it to you when I mentioned the quality control in the ER. But so from plants, what people did is borrow this system. So the AID is what's called the degron. So that's a signal. So that's a domain that you can engineer and attach to a protein of interest, right? This target protein. And it's not going to interfere much with the function of the protein, very much like GFP, okay? Unless you co-express this protein tier 1. So tier 1 is also a protein from plants. So this is all borrowed from plants, right? And normally tier 1 does not do anything except it recruits these other proteins, which is basically E3 ubiquitin ligase complex. So it's going to tag the protein for degradation, right? So you have tier 1 that you express in cells and is going to recruit the endogenous system to ubiquitinylate proteins. And it's not going to do anything until you add auxin. So auxin is a plant hormone. It's a small molecule, and it's not present in animals. So if you tag your protein of interest with this AID and express tier 1, nothing's going to happen until you provide auxin. Sorry. And what auxin will do is bring together tier 1 and AID specifically. That means now you have recruited the whole ubiquitinylation complex to your protein of interest via the AID, and now it's going to be sent for degradation. So this is one way to control the time at which you're going to degrade the protein. So that means the cells can express the AID tagged protein. It's going to work normally for the most part, except when you come in and treat with auxin, it's going to send everybody to degradation. And not everybody, just this protein. Okay, so now you can have a better control of the timing. And I'm going to just illustrate an example where it's done. So here in C. elegans worms, so these worms, they express both tier 1, right? So that's the protein you need to recruit the degradation complex, tagged with ruby, a red fluorescent protein. And it's co-expressing this dynein heavy chain. So what's dynein heavy chain? Molecular motor on which kind of elements? Microtubules, which direction? Minus n, very good, very good. So let me ask you a question. If you make a mutant for that protein, do you think you can get a worm? If you make a mutant worm of dynein heavy chain, do you think you can get the embryo to develop into a worm? Probably not. Exactly. There will be a lot of problem with mitosis and so on. So that's the interest of this kind of approach, which is you're not interfering with the protein function until a later time, right? Because with mutants, you would not be able to study the late function of a protein if it's required early. So this temporal control allows us to bypass early defects that you would get with a mutant. And so here, as a proof of principle, so this dynein heavy chain is fused to the degron, the AID, and to GFP, so you can see it fluoresce. And here in this group of worms, so it's the same group of worms in all the images, they all express both the tier 1 and this guy, except that only the worm on the left was not treated with auxin. Right, so they all have the same genotype, but only one was not treated. So what you're seeing very clearly is that the worms that have been treated with auxin, they have lost the fluorescence here. And they have lost it because that protein has been degraded altogether. Okay? And when you quantify that, only 10% is left, so that's very close to a null. Usually this amount of gene product left is not enough for function. So that's one way to use chemicals to modulate protein activity. Now let me show you another one, which is falling a bit out of grace because it uses this drug called rapamycin. And this drug was discovered to interfere with basically growth, and it targets a pathway which has now been called TOR, mTOR for mammalian target of rapamycin. So it's been well characterized since, and it controls ribosome biogenesis and cell growth in general, right? So that drug is not completely neutral, and this is one reason why this tool is falling, is being less used, but it's been very useful because people could determine that in the pathway there are proteins with at least these two domains, so-called FKBP and FRB. So they come from proteins in this pathway, and both can bind this drug rapamycin, which is shown here. Right? FRB binds on this side, FKBP binds on the other side. So here you have the principle of two proteins that will not normally bind until you provide a compound. Right? So you can decide when you provide a compound, and upon treating the cells with rapamycin, now you create this complex here, so you can associate the two proteins. And a lot of the cell biology we've seen involve dedicated protein-protein interactions. So if you can control that, you can control the system quite well. So let me show you one really cool example. Almost, as a proof of principle. So here people took KIF3, so it's a kinesin. What's kinesin? Yeah, that's a molecular motor that works on microtubule towards the plus end. Right? So as we've seen, it has these globular heads, the motor heads, they hydrolyze GTP, they bind to microtubule. They are the business end of the motor. They tend to be attached to these coil domains for dimerization. And here what they did is engineer it to have it bound to GFP, so it can be visualized, and to FRB, right? One of the two domains that can be used with rapamycin. So now, this experiment is completely artificial, okay? So you create this motor. So that protein is going to be able to walk on microtubule, and somehow you have FRB. So if you treat with rapamycin, you're going to be able to recruit FKBP, okay? And the protein used that's bound to FKBP here is EB1. What's EB1? I can't understand if you all remember that.
Plus tips.
Plus tips, that's right. So it's a plus tip binding protein, right? So now it's potentially really interesting what they're going to try to do here, because they are going to, upon upon treating with rapamycin, cause association of this EB1 protein with the FKBP and the molecular motor, FKBP and FRB, and the motor kinesin. So what are they trying to do here? Right? It's kind of weird, because you have two different kinds of microtubule binding proteins. So let me show you the movie. And you tell me what you think is happening. You see this? So what do you think is happening? There's no branch. So, right, remember, the two proteins, one is EB1, one is kinesin. Yeah, so what you have here is you have the tip of this microtubule, and it seems to be tethered to this other microtubule and walking towards the plus end. So if you had to interpret what's going on is that you have the EB1 protein binding to the tip of that microtubule on one hand, but then upon rapamycin treatment, it's now coupled with a kinesin, which is walking on the other microtubule. Okay, you guys are hard to please.
My phone's going back and forth.
Well, because it falls off. Yeah. And also because the movie loops. Yeah, so that's the interpretation. But let's say, so at the final exam, that's one thing I could ask you. If I give you the thing and I show you the phenotype to explain what's going on, right? So the idea is that indeed you have the EB1 protein tethering the complex to the plus tip of this microtubule, but then because it's also attached to a kinesin, it allows it to walk, and now you've effectively tethered the tip of a microtubule to the shaft of another microtubule. Okay, so this is all nice, but somehow what are we still lacking with these drug treatments? Auxin or space, that's right. We are lacking spatial resolution. We are treating the whole cell. it's good to be able to choose the timing, but we're treating the whole cell. So what could we use to focus on a particular location in the cell? Yeah, targeting sequences, but we would still express it. I mean, that's a creative idea. I'm thinking of a more experimental way. Light. Because we can shine light not only when we want, but where we want. Okay? So now let me tell you about some optogenetic tools. So a lot of what I've showed you so far involved protein-protein interactions, right? Controlled protein-protein interactions. And so the first class of optogenetic tools I'm going to show you also borrow from plant biology, where, you know, upon shining light on these proteins, you can cause them to heterodimerize or instead to dissociate. So you control protein-protein interactions via light. So, and there's a whole lot of systems. So they span the wavelength of light you can use. So this goes from UV, of course, to blue, green, yellow, orange, and even some red and far red here. So typically, I'm just going to show you some examples. So we've seen this one, the LOV domain, when I showed you photoactivatable rack, right? When we could force protrusive activity. So that was using this love domain, and I said the default configuration is closed onto itself, so you can have it trap the rack, but when you shine blue light on it, now it opens up and now rack can now be active. That was the example the other day. Another system that works in the blue wavelength is this Cry2Sib, which initially comes from, they all come from plant initially. This one has been also engineered to improve now. But now in this case, you have a different logic. So these are two different proteins, and when you shine light, they come together. So this is very useful. I'm going to show you some examples. And I'm just going to highlight this one here, PHI-B-PIF, which is really interesting because it's activated by far red. So basically, you can image your system and so on with blue light, and you're not activating the system until you shine far red light. Okay, so the fact that they all have wavelength specificity is useful to be able to combine imaging, so imaging of these biosensors, for example, with manipulating using these optogenetic tools. Okay, so again, so the way in which you would use those, right? So we saw the LOV example. It's closed onto itself in the dark or until you shine blue light. And when you do, you free the target protein. That's what TP stands for. And you can have active GTPase. We saw that with RAC. And we're going to see an example of using Cry2Sib. So in this case, it's dimerization that's induced by light. And so if you tether one of the two, for example, to the plasma membrane, as I show you, and the other protein is diffusing in the cytosol, so there's not enough at the plasma membrane to have any effect. Upon shining light, you can recruit it and have an effect. So what kind of protein would you want to regulate at the plasma membrane, for example? That's not an easy question. GEFs or gaps, right? Because what would that do? I'm just thinking about an exam question right now. That's right. So if you built it in this way, you can shine light in a particular location, recruit a GEF or a gap, and locally activate or inhibit a particular row GTPase, right? So that's fair game. All right, so one example showed here where they do exactly that. So you tether SIB-N to the membrane with GFP. So how could you tether a protein to the membrane? Yeah, so you can have a lipid modification like the small GTPases, an acylation signal, or you can have a transmembrane domain, and so on, right? So it's relatively straightforward. But then this protein here is not at the membrane. So you see here the GFP, the protein accumulates at the plasma membrane. This one here, the Cry2M cherry, so M cherry is red, is normally in the cytosol, that's before excitation. But then when you shine blue light, this it recruits this protein here, the cherry, the Cry2M cherry, to the membrane, right? So, and that's very efficient. So when people quantify this, so here again you have the membrane protein labeled with GFP. When you look at the M cherry protein, before the excitation, so that's 0.3 seconds before, it's diffused in the cytosol, and shortly after excitation, it accumulates at the membrane very quickly. So if you quantify the cytosolic fluorescence versus the membrane fluorescence, you get a lot of cytosolic fraction, and then upon shining light, the cytosolic fraction goes down, the membrane fraction goes up. Right? So it's very quick and very local. Now with the microscopes, you can do this. You know, if you have a good objective, you can have your cell, you know, and decide to activate shine light only in one place. So you control both space and time. And if you have a multiphoton microscope, like we do in this department, you can even control not just the XY position, but even the Z position. So activating a specific volume in the sample. So now, why would you want to do that? I gave you this theoretical example, and this has been done to recruit a gap to block, say, Rho, or a gap and activate. You can also do it to regulate gene expression. So in this paper, they did exactly that. So there's the famous GAL4 UAS system. So hopefully you've heard of this. So GAL4 is a transcription factor. There's a DNA binding domain and an activation domain. And when it binds to the UAS sequence, it can activate the gene. It's a classic, comes from yeast, but it's been used in flies a lot. And you can split it in a way that you have the DNA binding domain on one hand and the activation domain on the other. So each one of those alone is not active. Only when they come together, you have a functional transcription factor. Now you can control that functionality if you split and associate with Cry2SIB. And now only upon shining blue light, you reassociate this whole complex, and now gene expression can take off. Okay, so this experiment was done in the yeast. All right, so we have a few more minutes. So let me show you another. So all of the, yes?
Why does the gene expression have to depend on space?
Oh yeah, no. In this case, it doesn't. Yeah. So well, you could say it would depend on space if you're in so in yeast it's irrelevant. But if you were in a metazoan, you could activate only in some cells and not others.
Right.
So you would still have spatial control might still be interesting.
So there's basically no need for the chemicals, and then you can only control everything with the light. Like why
Well, it's partially historical, but chemicals are also easier to if you want to if you don't care about spatial control, and you mostly want temporal control, but you want to be able to treat a lot of samples, chemicals might be easier. Okay. All right, so let me show you one last example before seeing, I think I might just skip the synthetic biology part. One last example of a very powerful and famous optogenetic control tool. So famous it made it into your textbook, because you've noticed a lot of the examples I gave you were from primary literature. That doesn't mean it's optional. But so this one is so good, it made it into the textbook. So initially, people were looking at these algae, Chlamydomonas reinhardtii. And in a way, that's another shout out to basic research using, curiosity-driven, using all kinds of organisms, because you end up finding things that are very useful biomedically. Which other example of that can you think of? CRISPR, yeah, that's the famous one. So in this case, from this algae, so people have used it to study the flagella, microtubules, and so on, and its ability to respond to light. And they found this so-called channel rhodopsin here. So as the name indicates, it has two main properties. It's a channel, right? So it can control, I mean, really the function is in the name. So it's a sodium channel, so it can regulate the import of sodium. And you've seen with Claude, when you activate neurons, one of the things that changes is the sodium concentration inside the neuron goes up, right, when these channels open. So it's very important to control the transport of sodium across the plasma membrane. But this one is also rhodopsin, so it has this small molecule associated with the protein. So this is a channel, right? So that's a transmembrane protein, but it just happens to have this whole transretinal molecule associated to it, which changes conformation when you shine light on it, and therefore changes the conformation of the channel. Bottom line, this channel opens when you shine light on it, right? So that's very powerful because that means you can now control the membrane potential just with light. Yes? Oh, no, it is light gated. So in the algae, so in a way, it's not naturally found in all organisms, right? It comes from this algae, and it's been essentially repurposed for experimental purposes in mammalian cells and so on, as we'll see. So indeed, when you shine blue light on cells expressing this channel rhodopsin, you can now create spikes or action potentials when you have blue light, right? So this is pretty amazing because this is now giving you the option, and I don't know, did Claude tell you about this? Or did he give you examples of that? Because you can basically control neuronal activity by shining light on them, right? So, and all you need is to do two things: is express the channel rhodopsin and then shine light. So people are doing a lot of that, and here I'm going to show you the example that's in the textbook. So people express this channel rhodopsin in this specific part of the mouse brain. I forgot what SFO stands for. It is not the airport in San Francisco, but it's the organ that controls thirst, right? And basically, it's kind of top-down control. The animal, if you activate neurons in this region, will want to drink, right? So that was the idea. So here they show that you can express channel rhodopsin in these neurons. It's fused to YFP, so you can see it. And you can patch clamp, so you can measure the action potentials. And then so it is a bit invasive still, because you still have to come in with a blue light, right? So in the brain of the animal. And when you shine pulses of this blue light, you get action potentials, right? So effectively, now you're controlling neuronal activity in the brain of a live mouse. So then the question is that mouse going to want to drink when you shine blue light on its brain? And the answer is yes, right? So this summary experiment is also a very good illustration of a good experimental design with the right controls, okay? Because you want to make sure the response, so the drinking response here is only happening when the mouse is infected. So what infected means here is because the way the transgene is delivered to the neurons is via viral infection. Okay, it's not a transgenic mouse in this particular case. You can inject virus, they will infect cells, and therefore make them transgenics. Okay? So if you don't infect the cells, you can shine blue light, nothing happens, because there's no channel rhodopsin. If you infect the cells, there's channel rhodopsin, but if you don't shine light, nothing happens, because you're not activating the channel rhodopsin. Only when you have infected the mice and you shine blue light, now you get a very strong response where the mouse can be, willing to drink.
Can you do that in any cell?
Well, presumably, yeah.
So you can control someone's mind with light.
You can only turn it on, you can't turn it off.
Oh, true.
What, sorry?
Can you turn it off? Like, can you turn on off the neuron?
So this is a great question, and yes. So there are now a whole toolkit a whole optogenetic toolkit where you can shine light and inhibit neuronal function. Sorry?
Do you use that for seizures or
You know, transgenic humans is not. I think that's the FDA bottom line here is
No,
that
they just did immune cell research with cancer.
Yeah. Yeah, so yes, I saw this came out today, CRISPR-based for immunotherapy. But yeah, in principle, in mouse and in model organisms, it's been used a lot. In humans for for
Just chronic
medical purposes,
Yeah.
yeah, I don't think we're there yet.
This is the next.
Hmm?
This is the next dystopian online.
Well, no, I mean it's a good dystopian plotline. No, I agree with that. So talking about dystopia, I'm just gonna very quickly show you what I wanted to show you for the last part, very briefly. So this is mostly from this paper where two papers. One with this guy Craig Venter, which I removed this picture from my presentation because we didn't want to emphasize people too much in the course, and I think you guys should, there is a lot of starification of the science enterprise, and I don't think it's necessarily a good thing. So let's just focus on the work and the data. So the idea is that cells are largely modular, right? So you can identify bits and pieces of a cell, and it's the sum is more than, so the cell is more than the sum of the parts, but you can still find the parts and see how they work together, right? So this is just to illustrate that in this paper, focusing on a very simple cell, this Mycobacterium, they identified a bunch of modules, functional modules, and the genes that go with it, right? So in other words, they were trying to create a complete model of a cell from first principles. And when I say first principles, I don't mean atoms, I mean genes, okay? So how do you get a cell functioning from genes? So just illustrating this, you have a lot of features, right? Metabolism, protein function and synthesis, RNA, DNA, and you can break those down into these sort of modules such as translation, and I'm not going to go through all of them. And so they try to computationally test if, what's enough for a cell to live, right? Trying to find the minimum component of a cell. And then what Craig Venter ended up doing, and what I wanted to finish with, is this idea that, quoting Feynman, that what you cannot create, you cannot understand, or you do not understand. It's given a lot of credit, but the guy, I decided to remove his picture because, anyway. In a long story short, in the Me Too era, he would probably not have so much fame. So what Craig Venter did, and that's pretty remarkable, is create, so it's Promethean also. Do you guys know Prometheus? Greek mythology? Come on, tell me you've heard of this. What? The father of liver regeneration. He had an eagle coming. And only because he wanted to give information to humans. I think this is the fate of Julian Assange. Shit, it's on record. Okay, so what they did is use all this information, right, that they had about what is the minimum complement of genes you need for a cell to fulfill its life, at least fulfill a whole life cycle. And so they went through these, so in this paper published in Science now a few years ago, they went through these iterative rounds of designing and synthesizing. So the cost of synthesis of DNA is also going down as the cost of sequencing. And you have people like the medical school, Jeff Booker, for example, is synthesizing entire yeast chromosomes, so very large pieces of DNA. So here in this paper, they basically synthesized an entire genome for a very simple organism, right? So very, very much a bottom-up approach. And they were essentially, and then cloning it using membrane-bound vector, and then growing it in a way that the endogenous chromosome of the host would be kicked out and then left with their artificial chromosome, right? So that's very Promethean kind of approach. But they ended up with this simple version, this so-called J. Craig Venter Institute, so another megalomaniac who needs his name everywhere. Synthesis 3.0, so 500 million base pair. So that's really small for a genome, okay? 473 genes, so that's like 1/10 of a bacterium, if you will. So very simple, and yet it lives, right? So this organism created from scratch, from a bottom-up approach, just by knowing what works in real organisms and trying to simplify it as much as possible, was still capable of replicating. It would not do well at all in the wild. It was in the lab, but still, that's a remarkable feat. So suggesting that now that the humans can create this, maybe they do understand something about biology. All right, and I'll stop here.